DNA is what life is built on. Each of our cells has DNA strands that measure 6 feet long in total if you'd uncoil them all. How do these strands fit into a 0.0002 inches long cell? Well, DNA structure allows it to organize in such a way that makes this possible. DNA structure, molecular structure of DNA. Study smarter. Do you want to see this and many more great infographics? Sign up now for free. Fig. 1. You are probably familiarized with the double helix structure of DNA. However, this is only one of the levels in which DNA structure is organized. Here, we are going to run through the structure of DNA. First, we'll focus on DNA nucleotide structure and complementary base pairing. Then, we'll move up to the molecular structure of DNA. We'll also describe how the structure of DNA is related to its function, including how a gene can code for proteins. In the end, we will discuss the history behind the discovery of DNA structure. Stop procrastinating with our Smart Planner features for DNA structure. Study Smarter's free web and mobile app. Get started now. DNA structure overview. DNA stands for deoxyribonucleic acid, and it is a polymer composed of many small monomer units called nucleotides. This polymer is made from 2 strands that are wrapped around each other in a twisting shape that we call a double helix. Fig. 1. To understand the DNA structure better, let's take just one of the strands and then untwist it. You'll note how the nucleotides form a chain. DNA structure diagram of DNA strand, molecular structure. Study smarter. Sign up now for free. 2. A single strand of DNA is a polymer, a long chain of smaller units called nucleotides. DNA nucleotide structure. As you can see in the diagram below, each DNA nucleotide structure consists of 3 different parts. On one side, we've got a negatively charged phosphate which is connected to a closed deoxyribose molecule, a 5-carbon sugar, which is itself bonded to a nitrogenous base. DNA structure diagram of DNA nucleotide structure. Study smarter. 3. The structure of DNA nucleotides, a deoxyribose sugar, a nitrogenous base, and a phosphate group. Every nucleotide has the same phosphate and sugar groups, but when it comes to the nitrogenous base, there are 4 different types, namely adenine, A, thymine, T, cytosine, C, and guanine, G. These 4 bases can be classified into 2 groups based on their structure. A and G have 2 rings and are called purines, while C and T only have 1 ring and are called pyrimidines. Since each nucleotide contains a nitrogenous base, there are effectively 4 different nucleotides in DNA, 1 type for each of the 4 different bases. If we take a closer look at the DNA strand, we can see how the nucleotides combine to form a polymer. Basically, the phosphate of one nucleotide is bonded to the deoxyribose sugar of the next nucleotide, and this process then keeps repeating for thousands of nucleotides. The sugars and phosphates form one long chain, which we call a sugar-phosphate backbone. The bonds between the sugar and phosphate groups are called phosphodiester bonds. As we mentioned before, the DNA molecule is composed of 2 polynucleotide strands. These 2 strands are held together by hydrogen bonds formed between pyrimidine and purine nitrogenous bases on opposite strands. Importantly, though, only complementary bases can pair with each other. So, A always has to pair with T, and C always has to pair with G. We call this concept complementary base pairing, and it allows us to figure out what the complementary sequence of a strand will be. For example, if we have a strand of DNA that reads a 5-TCA3, then we can use this sequence to work out what the sequence of bases on the complementary strand must be, because we know that G and C always pair together and A always pairs with T, so we can deduce that the first base on our complementary strand must be an A because that's complementary to T, then the second base must be a G because that's complementary to C, and so on. The sequence on the complementary strand would be 3-CAGT5. Since A always pairs with T and G always pairs with C, the proportion of a nucleotides in the DNA double helix is equal to that of T, and similarly, for C and G, their proportion in a DNA molecule is always equal to each other. Furthermore, there are always equal amounts of purine and pyrimidine bases in a DNA molecule. In other words, a GTC. A DNA segment has 140 T and 90 G nucleotides. What is the total number of nucleotides in this segment? Answer. If T a 140 and GC 90. T a CG 140 140 90 90 460. Create DNA structure notes faster than ever before. Study Smarter's free web and mobile app. Get started now. Hydrogen bonds between DNA nucleotides. Certain hydrogen atoms on one base can act as a hydrogen bond donor and form a relatively weak bond with a hydrogen bond acceptor. Specific oxygen or nitrogen atoms on another base A and T have one donor and one acceptor each, hence they form two hydrogen bonds between each other. On the other hand, C has one donor and two acceptors and G has one acceptor and two donors. Therefore, C and G can form three hydrogen bonds between each other. A hydrogen bond on its own is relatively weak, much weaker than a covalent bond, but when they are accumulated, they can be quite strong as a group. A DNA molecule can possess thousands to millions of base pairs, which would mean there would be thousands to millions of hydrogen bonds holding the two DNA strands together. Molecular structure of DNA. Now that we learned the structures of DNA nucleotides, we'll see how these form the molecular structure of DNA. If you had noticed, the DNA sequences in the last section had two numbers on either side of them, 5 and 3. You may be wondering what they mean. Well, as we said, the DNA molecule is a double helix composed of two strands that are paired together by hydrogen bonds formed between complementary bases. And we said that the DNA strands have a sugar phosphate backbone that holds the nucleotides together. DNA structure diagram of molecular structure of DNA. Study Smarter. 4. The molecular structure of DNA consists of two strands forming a double helix. Now, if we look closely at a DNA strand, we can see that the two ends of a sugar phosphate backbone are not the same. At one end, you have the ribose sugar as the last group, while at the other end, the last group must be a phosphate group. We take the ribose sugar group as the beginning of the strand and mark it with 5 by scientific convention, and you must have guessed it, the other end that finishes with a phosphate group is marked with 3. Now, if you are wondering why that is important, well, the two complementary strands in a DNA double helix are, in fact, in the opposite direction of each other. This means that if one strand is running 5 to 3, the other strand would be 3 to 5. So if we use the DNA sequence that we used in the last paragraph, the two strands would look like this: 5-CAGT-CA3, 3-CAGT-5. The DNA double helix is antiparallel, meaning that the two parallel strands in a DNA double helix run in opposite directions regarding each other. This feature is important because DNA polymerase, the enzyme that makes new DNA strands, can only make new strands in the 5 to 3 direction. This creates quite a bit of challenge, especially for DNA replication in eukaryotes, but they have pretty amazing ways of overcoming this challenge. Find out more about how eukaryotes overcome these challenges in the A-level DNA replication article. The DNA molecule is very long, therefore, it needs to be highly condensed to be able to fit inside a cell. The complex of a DNA molecule and packaging proteins called histones is called a chromosome. Create and find the best flashcards for DNA structure. DNA structure and function. Like everything in biology, DNA structure and function are tightly related. The characteristics of the DNA molecule structure are tailored for its main function, which is to direct protein synthesis, the key molecules in the cells. They perform various essential functions such as catalyzing biological reactions as enzymes, providing structural support for cells and tissues, acting as signaling agents, and many more. DNA structure and function. Study Smarter. 5. DNA structure and function. The sequence of nucleotides in the DNA codes for the sequence of amino acids in a protein. Proteins are biomolecules made up of one or more polymers of monomers known as amino acids. The genetic code. You might have already heard of the term genetic code. It refers to the sequence of bases that code for an amino acid. Amino acids are the building blocks of proteins. As mentioned earlier, proteins are a huge family of biomolecules that do most of the work in living organisms. Cells need to be able to synthesize a plethora of proteins to perform their functions. The DNA sequence, or more specifically the DNA sequence in a gene, dictates the sequence of amino acids for making proteins. Genes are DNA sequence that encodes the creation of a gene product, which can be either just RNA or a protein. In order to do this, each group of 3 bases, called a triplet or a codon, codes for a specific amino acid. For example, a G would code for one amino acid, called serine, while GCT, called alanine, codes for a different one. We dive further into the genetic code in the gene expression article. Also, check out the protein synthesis article to learn how proteins are built. Everything you'll need for your studies in one place.